Alright, next we are going to look at the innate or non-specific immune system. Okay, so the innate immune system is the first line of defense of the immune system. It serves to first prevent infection, right? Prevention is always better than cure. So it prevents infection, but if infection does occur, if the pathogens does enter the body, then it also provides the early response to such infections. Okay, so there are different mechanisms to the innate immune system. We have the external mechanisms, which involve the external barrier, as well as the internal mechanisms once the pathogens have breached this external barrier. Okay, the features of the immune system, the innate immune system, is that it is always active. The cells do not need to be activated. They are always in the active or near active state, so they are able to function very quickly to provide the initial early response to infection. As mentioned from the name also, right, of the immune system, it is non-specific, and there is no immunological memory, right? So there's no enhancement or no improvement in the response, even if the immune cells encounter the same pathogen again, right? Okay, so firstly, for the external mechanism, right, the barriers, where we have both the physical and chemical barriers. Right, so for the physical barrier, right, it's written as mechanical here, but it's physical lah, physical barrier, right? Physical. So it involves mostly the skin, the mucous membranes that covers the internal surfaces like our respiratory tract, our gastrointestinal tract. These are like inside the body but exposed to the outside. The eyes as well, right? Okay, so if you need more details, right, these are some of the mechanical or physical barriers for the specific parts of the body, but collectively lah, this is just the skin and the mucous membranes, which basically blocks, right, the entry of the pathogens. Okay, then we have the chemical barriers as well, which involves again the mucus of the mucous membrane, which is a chemical, right, that traps any pathogens. And also our skin is acidic slightly, right, which inhibits the growth of pathogenic bacteria. And some other chemicals would be enzymes, like lysozyme, right, in our tears and saliva, that can destroy bacteria. If you remember the bacterial topic, right, lysozyme can break down the peptidoglycan cell wall of the bacteria. And also other secretions, right, from the cells of the skin, for example, would be this peptide called defensins, which can create channels, right, on the bacterial surface and cause them to lyse as a result. Right, so such molecules that can kill the pathogens are called microcidal molecules. Okay, so again, the different specific chemical barriers lah, in the different parts of the body. But you don't need such details lah. Okay, and I'll add one last one, right? Besides the physical and chemical barriers, I will add a microbiological barrier as well. This is not included in the notes because the microbiological barrier involves commensal microbes or bacteria that naturally live on our skin and in our intestines. So these are called the commensal microbes. right? They are not really part of our immune system, right? Because they are not part of our cells. They are just bacteria or other microorganisms living on us and in us. So it's not included in the notes as an external mechanism, right? But then I thought it is important for us to know that besides our own immune system, we also have such microorganisms that help us. So they do help to defend against pathogens by limiting the growth of the pathogens, as they live on our skin and in our intestines. Okay. Right, so just a bit more on the gut bacteria, the good ones, right? How they can prevent infections. All right, I'll just leave you to read it. Okay, so let me just move on to the internal mechanism. Right? So when the pathogens do get into the body, right, and break through the physical and chemical barriers, our cells of the innate immune system would respond the fastest, because again, the immune cells of the innate immune systems are almost always active. So they don't require activation, they don't take the time to become activated, right? They can straight away function to clear the pathogens, all right? Unlike the adaptive immune system, the cells, right, of the innate immune system have receptors that recognize antigens on the pathogens. This one we have talked about in the previous video. And the innate immune system is non-specific, right, because the receptors on the cells of the innate immune system, they don't recognize very specific antigens, right? They recognize antigens that are found on many different pathogens. Right? So that's why it's described as molecules or antigens, right, associated with a broad class of antigens. Okay, so again, like maybe they recognize peptidoglycan, for example, right? Then they would know that it is a bacterial pathogen, but they don't know which kind of bacteria. Right? So that's how the recognition is non-specific, by recognizing antigens found on many different pathogens. All right, so again, example here, right? Peptidoglycan, or right, if it is a gram-negative bacteria, then we have the lipopolysaccharide that is found on the outer membrane, right? So whatever bacteria, as long as they are gram-positive, then they would have this very thick layer of peptidoglycan on their surface. That would be recognized by the cells of the innate immune system. So they would recognize that this is a bacterium, this is a gram-positive bacterium, but don't know which kind, right? Or if it is a gram-negative bacteria, then there will be this outer membrane with the lipopolysaccharide, right? So the innate immune cells would be able to recognize the bacterium, but not which type. Yeah, so that's why it's again a very broad, non-specific recognition. For example, for bacterium. Okay. All right. So let's move on. Right. So because it's receptors, right? So you learn cell signaling, right? So once the receptors bind to these antigens, then that will trigger the cell signaling cascade, right, to result in certain responses, which would then be involved in killing the pathogen. So some of these cellular responses would be the activation of certain genes involved in production of molecules needed to kill the pathogens, like microcidal molecules that we have talked about, as well as this protein called cytokines. Right, so cytokines are proteins that's secreted by immune cells or white blood cells that can signal other cells to perform certain functions, right? Examples would be if there are macrophages that have recognized and bind to certain bacteria, for example, then the macrophages can then secrete these cytokines that causes fever, and also to cause increased blood flow and increased permeability of the capillaries at the site of infection. All right. So that would then promote the inflammation. But inflammation is basically involved in inflammation is a result of the increased blood flow and increased permeability of the capillaries. Also, when we say inflame, right, we are really talking about certain swelling, reddening of the site of infection, right? And this reddening and swelling is really due to the increased blood flow and also the increased flow of or the increased leakage of blood at the site of infection. And that is due to the increased blood flow and increased permeability of the capillaries, which is a response to the release of cytokines. Okay, there is this increased blood flow and increased permeability of the capillaries to increase the recruitment of more immune cells to the site of infection, right? So remember that immune cells travel around the body through the circulatory system, right? So having more blood flow would increase the number of immune cells traveling to the site of infection, and when the permeability of the capillaries or any other blood vessels are increased, then the immune cells can more easily leave the circulatory system and enter the site of infection. That's how inflammation actually helps with fighting the infection, right? For fever, right, an increase in the body's temperature usually makes it a bit less optimum for the pathogens and slightly more optimum for the immune cells, usually lah. Okay, so here's the diagram, right, showing the increased permeability of the capillaries, right? So if you follow my cursor, this would be within the capillary, right? On the capillary walls, there are certain gaps actually, right? So with the cytokines, right, then there will be more gaps or bigger gaps. So that makes it easier for the immune cells here to leave the capillary and enter the tissue and enter the site of infection. Okay, so again, another diagram. Now this one you have in your notes, but I have added a bit more in a notation, right, to talk about how the cytokines, right, released by the macrophages here, and these are histamines and other kind of signaling molecules. They both serve similar functions to cause inflammation, to cause the blood vessels here, the capillary here, to have increased blood flow, and also to increase the permeability. Right, so as a result, right, the neutrophil, which is another immune cells, would be able to come in at the site of infection, and the macrophages and the neutrophils would together, right, be able to clear the pathogens via phagocytosis. All right. So having mentioned phagocytosis, right, I thought let's look at phagocytosis in a bit more details. Right. So phagocytosis is not really the same as endocytosis. Phagocytosis is it's an action that is quite specific to immune cells called phagocytes. Okay, so not all immune cells are able to phagocytose. So those that are able to phagocytose would be called phagocytes collectively. Okay, So for phagocytosis, right, there will be the ingesting of the pathogens via the formation of pseudopodia. These are like extension of the cell to surround the pathogens. Once within the cell, right, the pathogens are placed in phagosome, which fuses with the lysosome, so the hydrolytic enzymes can digest the pathogens, right? After that, the debris would be released, and certain fragments of the pathogens might remain in the cell for other functions, but most of them are released. Okay, so a bit more on the difference between phagocytosis and endocytosis, right? So endocytosis, as you have learned it before, it involves the invagination, right? The infolding of the cell surface membrane, right, to surround this substance to be taken in an endosome. For phagocytosis, like we mentioned just now, there is the formation of pseudopodia, right? The cell, right, or cell surface membrane here actually folds or extend out to surround the pathogen. Right? So the process is slightly different, and then to bring in the pathogen, right, in this phagosome. Okay. So for the immune cells, then it would be better for you to use terms like phagocytosis rather than endocytosis, right? A bit more specific for phagocytes, immune cells. Okay. All right. So now we will look at the different types of cells of the innate immune system. The first is neutrophils, which is the most abundant of the white blood cells or of the immune cells. They are very effective in killing pathogens because they release quite a number of different microcidal molecules. One of it is defensins that we have looked at earlier, but there are others as well. Okay, and they are a phagocyte, so they are able to phagocytose the pathogens as well. All right. They are quite mobile, as mentioned, right? They can easily leave the capillaries to enter the site of infection. They are very short-lived, and they usually would die after they have performed their function at the site of infection. So they form the pus, right? So when we have pus from infection, right, quite a high proportion of the component of the pus would be these dead neutrophils. And when we can detect an increase in number of neutrophils at the capillaries or at the peripheral blood, the blood at the surface of our body, right? That usually indicates infection, right? Very acute infection of or entry of bacterium and other pathogens. Yeah. So that would be neutrophils. Then we have the basophils, also known as mast cells, depending on whether it is in circulation or inside the tissues. All right. Whether it's a basophils or mast cells, they kind of serve the same function. They release histamines. So histamines are not cytokines, right? It's something else, another kind of signaling molecule, but the effect is quite similar. Histamines also causes increased blood flow and increased permeability of the capillaries, so therefore inflammation, right? And histamines or basophils and mast cells are more involved during allergic reactions. All right. So that's why if someone has an allergic reaction, one of the drugs to reduce the reactions is actually antihistamines, right? To remove the effect of histamines released by these basophils and mast cells. All right. Okay, so just a bit of some allergens, common ones, right? So this is a diagram showing you the function of the basophil, right? So when there is the entry of the allergens, not really pathogen, but allergens, right? Then the basophils would have receptors that recognize these allergens, bind to it, causes it to release histamines, and then histamines, right, causes increased blood flow, increased leakage of blood because of the increased permeability of the capillaries. So that causes this redness, swelling. All right. Okay, so before we move on, just wanted to share the plight of this person. You can Google this person, right? Bryn Duncan. Right? She has what we call the mast cell activation syndrome, in which her basophils and mast cells are overactive. Okay, so she has a very strong allergic reactions to many different types of allergens, some of which are not allergens to normal people. All right, so what you see on the right here is the doctors basically testing what she is allergic to. So you see all these different numbers, right, on different parts of her back, right? The doctors kind of introduce different stuffs, different molecules to test whether she is allergic to it. If she's allergic, right, just how we mentioned the functions of the basophils and mast cells, right? It causes inflammation and redness and swelling. So you see a lot of swelling here, right? So showing that she is actually allergic to, well, many different things, because you see like all the different numbers, each referring to a different molecule, causes swelling, some bigger, some smaller, but causes swelling lah. So she's allergic to, well, many different things because of these mast cell activation syndrome. All right. Okay, let's move on to the next one, eosinophils. These are immune cells, very specific. Not say specific, but they are more involved in immune response against parasitic worms. Okay, they are not specific to any particular type of worms, but if it is a worm, then it is usually these eosinophils that are involved. All right. So a diagram here, right, showing this parasitic worm inside the body, and you see it is a swarm by all these different cells. Most of them would be eosinophils. Some are neutrophils and macrophages as well, but mostly eosinophils lah. All right, another diagram here lah. Eosinophils with the receptors recognizing parasitic worms in general. Yeah. Okay, then we move on to monocytes and macrophages. Again, they are kind of the same thing, monocytes and macrophages, just depending on whether they are in circulation or inside the tissues. All right, so these are again phagocytes. They are able to ingest and remove the pathogens via phagocytosis. Another function of monocytes and macrophages is that they release cytokines for inflammation. Right, so the eosinophils don't do that. Neutrophils don't really release cytokines as well. It's mostly the monocytes and macrophages. And they also serve as antigen-presenting cells to activate the helper T cells of the adaptive immune system. Right, so we talk about what is this antigen presenting in the next lecture. Okay, just know that macrophages, monocytes are antigen presenting cells for now. Next one would be the dendritic cells. Right, these are found mostly in the lymph nodes. Lymph nodes are part of the lymphatic system that we have looked at earlier. Also, these are the lymph nodes, part of the lymphatic system. These are places where the lymph from different lymphatic vessels drain into. Right, so if the lymph is there are presence of any pathogens, then they would drain into these lymph nodes, which is densely packed with different immune cells as well as the dendritic cells. Right, so they will be able to detect then the presence of pathogens, right, at these lymph nodes. Okay, so the dendritic cells, right, also phagocytose the pathogens, and they also serve as antigen presenting cells. Okay, the dendritic cells, dendrites are like usually dendrites, right, is a term used for nerves to describe some of these projections of the nerve cells, right? But the dendritic cells also have such projections. They're not nerve cells, but they also have such projections, right, from their cell body. So that's why it's called dendritic cells. So the shape is quite unique, right? And we move on to the last cell type that we're going to look at for innate immune system, the natural killer cells. Quite a cool name. They are not phagocytes, so they do kill, but they don't kill pathogens by phagocytosis. They kill our own cells, but not the healthy ones, right? They kill the abnormal ones, those that are infected, say by a virus, for example, or those that are becoming cancerous, or any other abnormality. So our abnormal cells would be removed and killed by these natural killer cells. Right? So a diagram here to show the action of natural killer cells. This would be the natural killer cells, NK cells. Right, so let me just explain this diagram a bit. Right, so what's going to happen is these are our own body cells, right? The yellow color thing here is something that all our cells would have so that they can be recognized as our own body cells. Okay, so for the natural killer cells, as I jump to this part, if it is able to bind, right, to this yellow color thing, yellow color cell surface protein that show that this cell is a normal healthy cell, then as mentioned here, there is no action of the natural killer cells. So remember, we have talked about how the immune system, the immune cells, should be able to recognize self versus non-self. So this is one example how a receptor on the immune cells, natural killer cells in this example, would recognize cells of our own body by recognizing the presence of proteins that show that it is our own body's cells. Okay, but the natural killer cells also have another receptor that recognizes viral components or recognize another protein that a cell would make if it is infected. So here, right, as mentioned here, right, when this cell is infected by the red color virus, for example, it makes a protein, a stress signal, right, on its cell surface. So the natural killer cells can recognize the presence of this stress signal and know that, oh, this cell is infected. So therefore, kills the cell. All right. So this diagram, I hope, shows you not just the action of natural killer cells, but a bit of what we have talked about even in my previous video, right? How the immune cells have receptors to recognize our own cells, also recognize pathogens, and distinguish the two. All right? Yeah. Okay, so let's move on to another internal mechanisms, the complement system. These are just proteins made by the liver, and they can bind to surfaces of pathogens, which will result in the lysis of the pathogen, if it's a bacterium especially, or to facilitate phagocytosis, because once they bind to the pathogen, they kind of become a signal for the phagocytes, like macrophages, neutrophils, to basically recruit them, make it easier for them to come and bind to the pathogen. so that facilitates phagocytosis. Okay. All right, so these are the different mechanisms how the innate immune system protects us from infection early on.